Monoclonal antibody. Monoclonal antibody is a type of antibody that is produced by a single colony of cells. It means that antibody is generated by a single epitope. We know that epitope is an amino acid domain which is about 5 to 6 amino acid residues, and it has a binding position on a specific antigen. So the antibodies which are produced by us against a single epitopes are known as monoclonal antibodies. These monoclonal antibodies are very important for immunobiosensors. They have their importance in therapeutics and diagnostics as well. Monoclonal antibodies are produced by two different procedures. First is the Hybridoma technique and second is the A-site method. The Hybridoma technique is a combination of in vivo and in vitro method. It means that we use the animal and the lab facility as well for the production of monoclonal antibodies. But in case of A-site method, that is an in vivo method, it means that these antibodies are totally produced in a mouse or in an animal. Normally, a mouse or a rat is used for the production of monoclonal antibodies by this method. So just to refresh your memory, the production of monoclonal antibodies, first you inject an animal with an antigen to produce antibody. The cells from the spleens are separated and they are mixed with myeloma cells in the presence of polyethylene glycol. After fission, hybridoma cells are produced, which are cultured in a specific medium, and then monoclonal antibodies are harvested. A-site method is an in vivo method in which the monoclonal antibodies are produced by growing hybridoma cells within the peritoneal cavity of the animal. The hybridoma cells are injected into the mouse and these cells multiply there, and as a result in the abdomen, in the A-site fluid, these antibodies are produced. These antibodies have quite a higher concentration. You can see in this slide a comparison of the two techniques. In first technique, which is a hybridoma technique, we injected the animal with the antibody, and later after the production of antibody hybridomas, these hybridomas are injected into a mouse to produce the A-site method to produce monoclonal antibodies through the A-site method. Nowadays, this method is used for the production of monoclonal antibodies. The monoclonal antibodies have applications in diagnostics. They are used in radioimmunoassay, ELISA, and other laboratory methods. Pregnancy test is the one of the most important application of monoclonal antibodies. Monoclonal antibodies are produced against human chorionic gonadotropin hormone and are used to detect the pregnancy using the urine sample. The monoclonal antibodies are also used in cancer diagnostic, hormonal disorders, infectious diseases, and cardiac troponin test. The therapeutic applications of monoclonal antibodies include treatment of cancer, transplantation of bone marrow, transplantation of organ, autoimmune diseases, cardiovascular diseases, and infectious diseases. So in summary, for a immunobiosensor, monoclonal antibodies are very important. Monoclonal antibodies are produced by two different methods. The method, A-site method, is normally nowadays used for the production of monoclonal antibodies. These monoclonal antibodies have applications in therapeutics and diagnostics.